So when we talk about DNA, let's kind of get some overview of DNA, but recognizing this is not a biology course, we just need to understand the vocabulary. Although in some sense it makes more sense to start with the tiniest unit and build up to the bigger unit, I think it might be easier to start with the big unit and kind of dive down deeper and deeper and deeper. But it means I'm going to refer to vocabulary words that I won't get to till the next screen. So just bear with me. All right, so there are two types of DNA. There's also another kind of nucleic acid called RNA. We're not going to focus on it. But of the two types of DNA, we have nuclear DNA that is found in the nucleus of every cell. And then we have mitochondrial DNA that is found only in the mitochondria of a cell. But the mitochondrial DNA is only a small segment of the nuclear DNA, so let's focus mostly on nuclear DNA. It's much larger. There's many pieces of it, but I need like 13 whole chromosomes in order to get all of it. It's like a set of encyclopedias, and I need multiple volumes, multiple chromosomes. So it's a much larger molecule, and this is coding for pretty much all the proteins used in the cell. It has all the genetic information that gets passed down from your parents and ancestors. And then we have this mitochondrial DNA, as I said. It's just a small segment. It's not in a long chain. It's rather in a circle, and it's passed just from the mother, and it only codes for a few of the proteins. So in the past, when DNA was hard to get, hard to amplify, hard to type, mitochondrial DNA was sometimes useful. Nowadays, we are able to do nuclear DNA testing and typing so frequently that we really just focus on that. So human DNA, there's so much of it that we have to break it up into 13 different books, or 23 different books or chromosomes. So we have a set of 23 chromosomes. Each chromosome actually it's a pair of 2 chromosomes. And this is a long, long, long strand of DNA that's been coiled up into these, that's what each of these chromosomes are made of. So we see lots of these chromosomes. So here's my overall chromosome, which is this huge, very densely coiled strand. And if I get all the way down, I recognize what the individual tiny pieces of it are. So let's talk about a chromosome pair. Again, we have 23 pairs, but one particular chromosome consists of a pair of 2 different chromosomes. One chromosome is inherited from the mother, the other chromosome is inherited from the father. And so each half of that pair has the same loci and genes, but they might be different. So in other words, on one side, so both of them might have the gene for eye color, but one chromosome might be coding for green eyes and the other one brown eyes. And overall, both of them contribute to the fact that probably you'd have brown eyes. So this is just the overall idea of what is in a chromosome. We're going to focus down a little bit more on what we call these genes and loci. A loci or a loci is one little segment of a chromosome. So I'm kind of like showing different parts that might be loci on this particular chromosome. So it's one little part of it, and I can look at the analogous part on the other set of the pair. And what we're looking in general for is we've identified these loci because almost all of human DNA is identical. 99.9% is. So these loci are pointing out places where they are not identical. So a couple things why they might be not identical. One is that it's a gene, and it's coding for a type of protein, and different proteins have different traits. Another is it might be what we call a single nucleotide polymorphism. Again, polymorphism just means it's a segment of DNA where the code is not the same for every single person. And a loci is just talking about a location on a chromosome. And there are several types of loci. One is a gene, and those are the ones we focus on a lot. However, in forensics, we're going to focus on this third type of loci, which is tandem repeats. So it's a short section of DNA that has a repeating base sequence. We just see this pattern of repeating several times. over here on the right, I'm trying to sort of identify visually with a sample. So in other words, the green one might be one loci, and the red one might be another loci, and the purple one is another loci. And because I have two separate chromosomes paired together, again, one comes from mom, one comes from dad, I have two copies of each loci, and they might not be the same, or they might be the same. So we see that these would be things that would code for things, like some of these loci might be a gene. Maybe the green one's a gene, and maybe the red one is a short tandem repeat. We don't understand all of these loci. We just know that they are different. Genes we're having a better time understanding, but some of these short tandem repeats, we notice it, but we don't understand what it's doing. It's telling the gene, the cell something, and we don't understand it. So if I uncoil a chromosome, I'm going to see that it's composed of this long chain that it's twisted into something called a double helix. So it naturally has a like curly circling to it, and we call that a double helix. If we untwist the helix and look at it, we see that it's made up of what we call double-stranded DNA. So on this left side is one strand of DNA, and on the right side is another strand of DNA, and they're held together by like, it's kind of like a ladder, and they're held together by rungs, and these rungs are nitrogenous bases that are bonded together. And we call them base pairs because we've noticed that there is a pattern. There are 4 bases I'll talk about on the next slide, but adenine always pairs with thymine, or if I had thymine, it would pair with adenine, and guanine always pairs with cytosine, or if I had cytosine, it would pair with guanine. So this is what is holding the 2 strands together, is these base pairing. If I didn't have them held together, I would have 2 single strands, but the bases on them would be complementary if I were to pull it apart somehow. Okay, let's look at that specific, what we call DNA sequence. So everything we need, I can find by the sequence of bases on one strand. So those order will be, on the other strand will just be the complementary base pair. So I really only care about one strand, although they will exist together. And everything else is the same. So this DNA sequence is what we call our code. So when we want to identify one sequence of DNA versus one, gene or loci or STR compared to another, what we can do is list off the sequence of these bases. And that's what we call the code or the DNA sequence. Some of these sections, as we said, are recipes for building proteins. Other sections we don't understand, but is clearly telling the cells something important. Let's look at that DNA strand specifically. It's made of 3 different repeating units of nucleotides. Each nucleotide has 3 components to it. So this is going to be each nucleotide is consistent of a sugar and a phosphate and then a nitrogenous base. And again, we have 4 nitrogenous bases: adenine, guanine, cytosine, thymine. Luckily, they all start with different letters, so we just use the first letter of each to identify it. And so the sugars and the phosphates kind of make up the ladder side, the sides of the ladder. And again, these nitrogenous bases is the part that will connect one strand to another strand in the double-stranded DNA. So we have these vocabulary words you should be focusing on. What is a nucleotide? What's the 3 components of a nucleotide? What are the base pairs? And then understanding, once we get to the bigger structure, we have loci on chromosomes, and some of the loci are genes, and some are tandem repeats. It's also important that we need to be able to replicate the DNA. This is the master cookbook for everything that the cell needs to do, and so each cell needs to have its own copy of the DNA. Therefore, it has to have a way to copy itself accurately, and we call that DNA replication. Essentially, it begins with the original double-stranded DNA. Part of it unzips. As we said, each side determines what the other side needs to be, because the other side, the second strand of the double strand, has to be the complementary base pairs. So it unzips, and then a new strand is built in place by adding nucleotides of the complementary base. So once we're done, we end up with two identical copies of the original DNA, and when the cell divides, each copy would go into a different cell so that each cell has a copy of it. We will use something similar to this to amplify DNA that we find forensically using a technique known as PCR. A couple of times we do have these things called mutations, and it can get altered in a couple of different ways. You don't need to know the specifics about these or what's the difference between duplication and inversion. Just know that sometimes while it is being duplicated, a couple mistakes are made. We believe evolutionarily that this is how we have different kinds of DNA. This is why some people have blue eyes and some people have green eyes. That's how they've come. But so it differentiates individuals, but it's also can cause issues with specific individuals. Some things are fatal or cause diseases.